to talk about a game plan moving forward here. So, if this morning's lab was any indication, we're in pretty good shape in terms of our timing and stuff like that. This morning's lecture, we finished the respiratory system. We talked about CO2 transport in the blood and the importance of regulating CO2 because it's a major contributor to changes in blood pH. So what I wanted to do today is I wanted to go ahead and start talking about the kidneys and the urinary system. And we'll get through the majority of kidney anatomy today. And then on Friday, we'll have the opportunity to talk a little bit about renal physiology and stuff like that. So we're in pretty good shape right now. So the kidneys and the associated structures from a gross anatomical point of view are really pretty straightforward. When we take a look at the renal system on the models that you guys have in your benches or on a diagram like this, we see that you have two kidneys in your abdominal cavity. The kidneys, of course, are where urine is made. And then there's a tube that carries the urine from the kidney where it's made all the way down to the urinary bladder. Those tubes are called your ureters. Urinary bladder is going to store urine until we need to get rid of it. And then when we do get rid of it or empty our bladder, there's a tube that carries it out of the body. That tube is called the urethra. Okay? In males, the urethra is shared with the reproductive tract. Both semen and urine leave out of the urethra. For the females, the urethra and the reproductive tract are completely separated from each other. So, a little bit different there, but nevertheless, everybody has the urethra. So, from a gross anatomical point of view, this is really the majority of the renal system anatomy. You have two kidneys, two ureters, a urinary bladder, and a urethra that carries the urine out of the body. So it's relatively simple in terms of its anatomy. The kidneys are not terribly large organs at all. The models that we have scattered about kind of show the kidneys like these little, a little bit smaller than your fist, and that's about how big they are. They only account for about 0.4% of your body mass, so they're really, really small compared to other organs that we have in our body, which make up a much larger percentage of our body mass. So, it's a fairly compact organ system, and like I said, from sort of a gross anatomical point of view, there's really not a whole lot there. All right. Let's go ahead and talk a little bit about the kidneys, and let's relate a little bit of this to our urine and our own observations about our urine. Right? I joked with you guys a week or so ago when we've been together a few weeks now. We can talk about mucus. We can talk about urine too. So think about your urine really quick. And what are some of your observations about your own urine? What do we see? Specifically, like what do we see like day to day, change to change, things like that? What's that? The color, it's yellow, right? Yeah, indeed it is. It's a pigmented waste. All of our waste is pigmented, so is our feces, okay? So it's a pigmented waste. What else do we see? Do you always pee the same amount? No, you don't, right? When do you pee more? The morning, okay. The morning, for sure. When you first get up, right? Yeah, for sure. What else?
After you drink a lot.
After you've had a lot to drink, right? Right. I noticed for me when I drink more water or fluid, I typically urinate a lot more. Whereas when I'm caffeinated and I don't drink as much, I urinate a lot less, right? So there's this kind of correlation between the amount of fluid that I'm drinking and the board right now, right? You guys have, I don't remember if we talked about this in the first week of class, but if you had me in 50A, I know we talked about this. You have 3 primary fluid spaces in your body. Do you guys know what those primary spaces where fluid is found are? I bet you do. You just haven't thought about it this way. So one of the places I find fluid is intracellular fluid, right? The fluid inside of my cells. Okay. Next one is extracellular fluid, right? Okay. So around my cells and inside of my cells. And then where's the last place? We just finished with this organ system, by the way, and before our last exam.
The heart.
Okay. What's the fluid?
The serous fluid?
Huh? No? That's a very small amount. That would be extracellular fluid. Blood plasma, yeah, blood plasma, right? Blood plasma is our last one, right? So inside of your blood vessels, okay, inside of your cells, and then around your cells. Those 3 compartments are your primary fluid spaces, okay? I know we don't normally think about it that way, but if you look at all of your fluid, that's where it's all found, is in one of those 3 places. So when you drink a lot of water, right, when I drink a lot of water, it has to go somewhere, right? It has to go somewhere. So what it does is it gets absorbed and it starts to fill up. First it gets absorbed into my blood, right? So my blood plasma volume goes up. What's that going to do to my blood pressure? It's going to go up, right? Okay? And then if it gets moved out of my blood plasma, it gets moved into my extracellular fluid, right? So my extracellular fluid volume is going to go up. What do we know about extracellular fluid? Is water the only thing that we care about in extracellular fluid? Definitely not, right? You have things like sodium and potassium in your extracellular fluid that are really important for action potentials, right? If those concentrations gets messed up, then we have issues with resting membrane potential and all sorts of things like that, right? So if I add more water to that fluid, that is going to mess up the concentration of sodium, for example, right? So what we have to do is we have to maintain a relatively constant amount of body water to regulate blood pressure, to regulate extracellular fluid volume, okay? So when you drink a lot more water, what you have to do is you have to get rid of a lot more water to maintain a relatively constant amount of body fluid, okay? So you guys have already seen this, but you have to maintain homeostasis of your fluid volumes inside of your body, or else we're going to run into problems. Okay. From blood pressure points of view, from action potential points of view, and things like that. You know, one of the things that we can do to kind of illustrate these points is we can look to other animals. I'm a comparative physiologist by training, so I usually think about things in non-human examples. And I think we all probably need to look at cute animals today, right? So, does anybody know what this is? What's this a picture of? If you've ever been camping in Joshua Tree, you probably have seen one. Maybe go for a hike in Sycamore Canyon, like after sunset or before sunrise, you can see them. You know what they are?
A rodent?
It is a rodent. Good job. Okay. You guys are on the right track. It's on the ground, so that's a good sign.
I'm like super sure that's
this is a kangaroo rat. This is a kangaroo rat. These are one of my favorite animals. I love kangaroo rats. I think they're just the most charming little creatures. Right? They're adorable, as you can see, and they've got big sideshow bob feet, and they hop around like kangaroos, which is how they get their names. Right? They don't lose a lot of big jump. And they're really good at it. They have big long tails which you can't see in this photo, but they have big long tails that help the balance and stuff like that. And they move around after dark. They're heavily nocturnal, but they move around and then they look for seeds and stuff like that to eat, and then they go into their underground burrows during the day to sleep. They're really, really remarkable little creatures. And again, you don't have to go very far to see them. Just go for a hike in Sycamore Canyon and you can see them after dark. They're pretty easy to find.
As soon as it gets dark, if you have your flashlight, you'll see them scurrying across the trail in front of you and stuff like that. There's lots of different species, but we have a lot of them in Southern California. So unless you're a mammalogist and a good one, it's hard to tell the difference between species. You have to like count toes and stuff like that. And that's kind of challenging. But nevertheless, they're really, really charming little creatures in a lot of ways. But one of the things that makes them really amazing as it relates to kidney physiology is the fact that we think that for basically the entirety of their lives, they don't actually ever take a drink of water. Okay? They're really well adapted to living in desert ecosystems. Again, you can see them here in Riverside, but you can also go out to Joshua Tree and stuff like that, and you can find them pretty easily as well. But it's believed that they don't ever take a drink of water in their entire lives. Okay? So that tells us that they're really good at utilizing the water that they do get, right? You do get a surprising amount of water from the food that you eat. Okay, you probably get about a half a liter of water a day from the food that you eat. That's about half of a water bottle, right? They eat seeds which have some water in them. Okay, so there is some water in those seeds that they eat, and apparently they get enough from those seeds to be able to survive. Okay, but what that means is that if they aren't drinking, then they can't be wasting urine, right? They can't be peeing all over the place or anything like that, because that would be a huge waste of water. So, you're dehydrated, you know that you haven't had a drink of water all day long, and you finally, at the end of the day, go pee, right? Your pee is really concentrated, really yellow and stinky and concentrated. There's not very much of it, right? Just a little trickle. Again, we've been together 12 weeks. We can talk about our urine, right? We've all been there. We don't have to be ashamed of it, right? We can talk about it. But it's you know, you're dehydrated. You know what that's like, right? These guys, when they urinate, are not only concentrating their urine, they're producing a really small amount of it. In fact, it's only a drop or two at a time that they urinate. So they're able to concentrate their urine, okay? We'll talk a little bit more about urine concentration and the way that we measure it on Friday. But suffice it to say, think about your most concentrated dehydrated urine, right? They concentrate their urine about 10 times more than that. So it's really concentrated, and it's just a drop, right? So they're wasting very, very little water to help to adapt them to this desert ecosystem. Why do we think that they don't drink? Well, usually a lot of our evidence comes from captivity, but occasionally these animals are kept in captivity for various reasons, and when they're given free access to water, they do not take it. Okay, the most that they'll do is eat a piece of iceberg lettuce or something like that, but that's about it. They don't drink water, so they're really remarkable little creatures in a lot of different ways. Another one with a really cool kidney that is adapted to helping us to balance body water is the vampire bat. I don't have a picture of them, but vampire bats are probably things that you guys have all heard about. They aren't bloodsuckers per se, but they are what we call synchlivores, meaning that they drink or eat blood. Okay? They eat bugs and stuff like that too, but they do have blood as a part of their diet, and they have these incisors, these front teeth that are sharp and kind of stick out like this, right? And what they'll do is they'll land on like a cow or something like that, piece of livestock, and they'll make a really small incision on it. And then their saliva has an anticoagulant in it. So what they'll do is they'll mix a little bit of saliva into the wound that they just created, so the blood trickles out of that wound. And as it trickles out, what they'll do is they'll lap up the blood. Okay. Now, blood has a really high protein concentration, right? We talked about this when we talked about the circulatory system, and we looked at the composition of our blood, right? So blood has a really high protein concentration, and obviously the bats want that protein out of the blood that they're getting, right? But obviously there's a lot of water as well. Okay? Now, these vampire bats aren't very big. I mean, without their wings, their body's only about that big around. They're really, really tiny little things, right? And if you're a flying animal, you want to try to probably stay as light as possible, right? Because you're you're going to have to take off after you're done eating, right? But vampire bats will actually eat about 2 to 3 times their body weight in blood in one sitting, right? So when they drink, they're drinking a huge amount of blood, right? Like I said, they're doubling or tripling their body weight when they do this. So it makes taking off very, very difficult. If you triple your body weight and you're a flying animal, it's going to be really hard to take off, right? And remember, that's where they're safe, right? Eventually that cow is going to wake up and realize something is on it, trying to start flapping with its tail or something. That puts a little tiny creature like that at pretty great risk, right? So safety is up in the air. So they have to be able to take off when they're done eating. So what they do is to balance that body water out, they have these kidneys that work at an exceptionally fast rate. Right? When you're really well hydrated and you're drinking a lot of water, it feels like you're running to the bathroom every 15 minutes, right? Vampire bats' kidneys work at such a high rate that they're actually able to start to produce urine as they're drinking. Okay? So videos of them are really cool, and I tried to find one, I wasn't able to, but you'll actually see them start lapping blood from the wound, and within about 30 seconds to a minute, they start producing urine, and they literally urinate as they drink. So even though they're drinking 2 to 3 times their body weight, they're actually adding very little to their body weight because they're dumping all that water from the blood that they're drinking, and they're just absorbing the protein. So really, really remarkable little kidneys that vampire bats have. Really cool little animals. So animals adapt to their environment and to their diet in all sorts of different ways. And a lot of times that just simply means that we are needing to balance water. Okay? So regulation of that body water, both in the extracellular fluid and in the blood plasma, is really, really important. Another thing that our kidneys do is they can regulate the osmolarity of our blood plasma. Sometimes that involves dumping excess water. Okay, sometimes that means conserving water. Sometimes it means directly regulating ion concentrations as well. Okay, and we'll have some specific examples of some of these things. And then additionally, we use our kidneys to regulate blood pH as well. Now, this morning in lecture, we had spent some time talking about how the respiratory system, our lungs, work to regulate the pH of our blood. And simply by changing the rate and depth of your breath, you can regulate the pH of your blood, right? But that isn't the only place that we can run into issues with metabolism or with acid-base balance. If you're exercising, you're going to have lactic acid building up in your bloodstream, right? That's going to cause a shift in your pH balance. Cellular destruction can cause the release of all sorts of acids, like citric acid and stuff like that, and all sorts of things, right? So our kidneys also play a role in the regulation of blood pH, just like our lungs. We'll talk a little bit more about specifically what they do as we sort of get through some of this material. We probably won't get to that until Friday. Excretion of wastes, right? What sorts of wastes do we need to get rid of, right? Well, there's the byproducts of toxins for sure, right? When you are taking what we kind of generally call xenobiotics, but kind of familiarly we call these drugs, right? Where we have aspirin, right? Tylenol, all of these medications that we take, there are byproducts of those that we have to get rid of in our urine because that's the only place really to get rid of them. Okay. One of the students this morning when I asked about your observations of your urine, he said, "Yeah, he says, sometimes after I eat a certain thing or drink a certain thing, I notice a change in the smell, right? Think about things like asparagus, right? Like I really like asparagus. But I know that, no, I like asparagus. It's a good time in the summertime, right? But I know that after I eat asparagus, my pee really stinks, right? Because there's chemicals in that asparagus that I can only get rid of through my urine, right? After coffee, right? After you have coffee, sometimes your urine stinks after you have coffee, right? So there are chemicals in the things that we bring into our bodies that we can't metabolize, and we have to get rid of those through our urine, right? This is the reason why drug testing works, right? When we take illicit drugs, right? The byproducts of those end up in our urine as well, and that's the basis of drug testing, right? But that isn't the only type of waste that we have to get rid of. We also have the waste that we produce through our normal metabolism as well. When you guys learned about ATP synthesis and stuff like that in Bio 1, you looked at it from the point of view of a glucose molecule, right, sugar. But that isn't the only type of fuel that we can break down. Okay? We can also break down proteins. Okay? Proteins are made of amino acids, and amino acids get their name because they have these amino groups attached to them. Right? The NH2 is the amino group that we find on amino acids. Some nucleic acids also have amino groups attached to them as well. And when it comes to getting rid of this nitrogen here becomes a little bit problematic for us. We call it nitrogenous waste as a result of that. And there's a couple of different options that we have to get rid of this nitrogenous waste. What we typically do first is that NH2, that amino group, that NH2 is typically going to be converted almost immediately to NH3. Does anybody know what NH3 is? It's ammonia. Yeah, NH3 is ammonia. Okay. What do we know about ammonia? Other than you can clean your bathroom with it if you need to or something like that. It's really, really toxic, right? It's not good to have it build up. If you have something building up that's toxic inside of your body, one way to make it less toxic is to dilute the hell out of it, right? Dilute it with a lot of water, okay? So there are some animals that can actually excrete ammonia directly as a waste product, but the problem is to do that, you've got to do it with a ton of water, okay? The only animals that really works for is animals that live in water, because they can drink everything that they get rid of almost immediately, right? So fish, like what we have up here, these little fish that you see up here, these guys will actually get rid of ammonia directly without any further processing, but they live in water, and as a result, they can just drink what they just pee out. There's no real loss of water. Okay? That's fine for animals that live in water, but terrestrial animals like you and I don't have that much water available to us, right? I guess humans kind of do, but we also kind of don't, right? I mean, not everybody does. And definitely not all terrestrial animals do. Our kangaroo rats are a really good example of that, right? So water for terrestrial animals, animals that live on land, can be somewhat limiting. So what we have to do is we have to convert that ammonia to something that's less toxic, okay? Most terrestrial animals, especially mammals, will convert their nitrogenous waste to something called urea. Okay? Urea is way less toxic than ammonia, so as a result, it means that we can let it build up in our system a little bit, okay? Because it isn't as toxic to us. It's still not great, we still have to get rid of it, but it's way less toxic. We do still have to produce a liquid waste to get rid of it, though. We have to dilute it in some water to be able to get rid of it, which is probably why we produce urine in the first place, because it's a liquid waste. Okay? So we do find urea in our urine because we're getting rid of it that way. A third option that some animals use, mainly reptiles and their descendants, so like lizards and snakes and birds, as it happens, have evolved the ability to convert the nitrogenous waste to something that's not really toxic at all, but also not soluble, and that's uric acid. Okay? That uric acid from these animals is typically secreted as a white paste. You guys have definitely seen it. All you have to do is wash it on. It's inevitable that a bird will poop on it, and you'll see that uric acid, that white waste product coming out. It's not liquid. It's kind of pasty and gelatinous and kind of dries. It's usually mixed with poo because because birds and snakes and lizards only have one opening called the cloaca, so everything comes out the same opening, right? But it's a non-soluble paste of uric acid, and it comes out that way. Mammals, like you and I, do make a really small amount of uric acid, but normally it's not a problem for us, but we do make a very small amount. There are certain circumstances where that uric acid can crystallize in our bodies, and when it does do that, it lodges in joints, and those uric acid crystals can be excruciatingly painful when that happens. What's that called? Anybody know when you have buildup of uric acid? It's called gout. Okay, or gouty arthritis. Okay, gouty arthritis or gout is a buildup of uric acid. It crystallizes. Those crystals are like needle-shaped, and they lodge in our joints, and they become excruciatingly painful for people that have them. For some reason, and I don't have an explanation as to why, gout typically affects men, and for some reason, it typically affects the big toe, which is kind of a random. That's typically where those uric acid crystals find their way to. But apparently it's excruciatingly painful for people to have. I've had a swollen finger. My pointer finger's been swollen for like 6 months, and I don't really know why. I guess it's just a fun thing that I unlocked when I turned 45, right? It's like a new level in the video game of life. But I have a friend who's a rheumatologist at Loma Linda, and I asked her, I said, what's going on with my finger? And she says, do you have a family history of gout? And I said, no. So I went to the doctor, and I had my blood drawn, and there's no gout at all. All of the markers they use for it are perfectly fine. Same with rheumatoid arthritis. There's nothing, but I guess I just have a swollen finger, so I can bend this one all the way down. This one only goes about that far. So. Who knows? Like I said, you unlock new levels of things as you get old. It's kind of fun. That's all right. Okay. So anyway, no gout here. But it does tend to be genetic, which is why she asked if we had a family history of gout. But anyway, different ways of getting rid of that nitrogenous waste. But urea is the primary way that mammals get rid of it. And that's primarily what. Finally, we talked about the role that the kidneys play in red blood cell synthesis. We talked about that glycoprotein erythropoietin that the kidneys secrete that regulates the synthesis of red blood cells. And then our kidneys also play an important role in vitamin D3 metabolism. So lots of things going on in the kidneys. For such small organs, they're really, really remarkable little organs for sure. So what I want to do today is I want us to get familiar with the anatomy of the kidneys, and then what we'll do on Friday is we'll talk about how first we'll talk about how they make urine, okay? And then what we'll do is we'll talk about how we alter our urine to sort of address some of these major things, okay? So that's kind of where we're going to go on Friday. Because they're really, really remarkable little organs. And for something that contributes such a small amount to our body mass, they do a huge amount for our physiological homeostasis. So, one thing I do want to point out is that the kidneys have a really, really big blood supply. Okay, the renal arteries that supply blood to the kidneys are much bigger than we would expect based on the size of the organ. Okay. We would expect, maybe, if we didn't know anything else about our organs, you would expect maybe big organs to get a lot of blood and small organs to get a little bit of blood. Fair? It's a fair assumption that you can make, right? Turns out that that's not the case. I think we probably know that by now. But your kidneys are only about 0.4% of your body mass. This means that if you weigh 100 pounds, your kidneys only weigh 0.4 of a pound, right? So really, really small percentage. Yet they receive about 20 to 25% of all of your body's blood. That's a huge amount, right? So it's a big disproportionality that we see there, right? So really, really rich blood supply. This is also how we make urine, okay? We're going to take the things out of our blood that we don't want, and we're going to excrete in our urine. We're going to excrete those wastes and things like that. So what I think that does is it kind of speaks to the fact that we produce a lot of waste, right? A few times this semester I said that our metabolic rate is really, really high. And because we do consume so much in terms of energy and oxygen and things like that, it means that we're also producing a lot of waste, right? So carbon dioxide, just a big bag of hot air, right? That's too soon. So anyway, it's a lot of waste. Okay, so let's go ahead and talk a little bit about well, let me go through the blood supply really quickly. So like I said, about a quarter of our blood is filtered every single minute. High rate of waste production and stuff like that. So it's a pretty remarkable thing. So what I do want to do is I want to spend a little bit of time going through the anatomy of the kidney and kind of correlating this to some of the stuff that I have on the slide. But to do that, let's go back to the slide of the arteries here. Okay, so the arteries that are feeding this. You can see your renal artery right here, and we can see that it branches, and those blood vessels are the smallest on the outer edge of the kidney. So the kidney is a collection of blood vessels, but it also has these little tiny tubules called nephrons. And the nephrons are where the actual urine is made, right? And I want to spend some time talking about the structure of our nephrons and going through the anatomy of our kidney. So the kidney as a whole can be divided into two layers. So this is just a frontal section of the kidney, right? Kind of looking at it like we see right here. We just took a frontal section of the kidney. So the kidney itself can be divided into two layers. Whoops, let me use a little bit of a bigger pen. Right, something along those lines right there. The outer part of the kidney, where we see these really small blood vessels, that's called the cortex of the kidney. Okay? We have the cortex of the kidney right here. And then the inner part of the kidney, where there's more stuff, is the medulla of the kidney, right? We're going to look at both of those, and we're going to look at how these guys connect to each other. But again, you can see that the smallest blood vessels are up here in the cortex of the kidney. The bigger blood vessels are sort of at the center. I think if we've learned anything this semester, the action happens where the small blood vessels are at, right? That's one of the things that we know. Not much happens with big blood vessels. Those are just carrying blood. But the action is actually happening where we have the smaller blood vessels. So we got to start out here at the cortex, and then we'll kind of work our way inwards. As part of your lab today, you are going to be learning the names of all of the blood vessels that go into the kidney. But we know the renal artery, of course. These are what we call segmental arteries. These are what we call interlobar arteries. The kidney, the tubes in the kidney are organized into these things called renal pyramids. We'll kind of return to those in just a little while. But I want to look at these arteries that come up and over the top of each one of these renal pyramids, and then we can kind of go from there. So let me go to the board. Let's try to redraw some of this stuff. So again, we've got our kidney up here. Sort of divide out. Cortex and the medulla right here. What I want to do is I want to blow up this one area and take a little bit of a closer look. So I
I'm going to go ahead and I'm going to take a little section kind of right up here, and I'm going to take it over here and take a little bit of a closer look at it. To kind of correlate that to this diagram, we're going to kind of come over here, and we're going to look at this section of our kidney kind of right here. So we're going to kind of look at that. So one of the things that you'll notice is that I did include cortex and medulla in my little box. So what I'm going to do is I'm going to go ahead and I'm going to put the edge of my kidney kind of right here, and then I'm going to put that same little dotted line kind of right here, kind of dividing those two areas. So again, we've got our cortex right here, and we've got our medulla right here. You'll notice that if I take and I erased it, but my yellow line right here is sort of dividing my cortex and my medulla. Okay? You'll notice that one artery runs right along that dividing line, right at the top of this structure called the renal pyramid. Okay? So the arteries that we have right here are called arcuate arteries. Okay? These arteries are called arcuate arteries. They kind of are doing this. Called arcuate arteries. The way I always remember that is I remember that the arcuate artery arcs over the top of the renal pyramid. Okay, that's kind of how I remember that. Arcuate is that it arcs over the top of the renal pyramid. Okay? So that's an arcuate artery. You'll notice though that coming off of that arcuate artery are all of these little teeny tiny finger-like projections, right? They're all kind of coming off of it, right? Those are called cortical radial arteries. So it's called the cortical radial artery. Cortical radial artery. And I guess that name makes sense, right? Because they're radiating into the cortex of the kidney, right? So that's kind of how you can remember that. So it's a cortical radial artery. Now, we've learned that the arteries branch into smaller blood vessels called arterioles, right? So we need to look at a couple of arterioles here, right? So what we have is we have a situation that kind of looks like that. Let me draw it over here a little bit bigger. I know it's kind of small. So you've got your cortical radial artery kind of coming up right here like this, and then you have this blood vessels that kind of come up like this. Oh, it really sucks today. I'm sorry for that. Okay, so we have a situation that looks like that. I recreated my shitty drawing. Okay, so what we have here is we have the cortical radial artery, and it branches into these arterioles that we call afferent arterioles. Okay, kind of have a situation like that. Let's color it right there.
term afferent means going towards something. So what are we going towards? Well, what's happening is that this arteriole is branching into this ball of capillaries that I draw right here. I've been a little bit sloppy about that, right? But really what it is it's this capillary bed that just forms a little ball. Okay? A ball of capillaries is called the glomerulus. Which, in my opinion, is one of the more fun words in this class to say, right? Glomerulus. It's like a tongue twister, and there's a lot of syllables there. And then, of course, from there, we have a second arteriole that leads away. This is called the efferent arteriole. There we go. So there's our efferent arteriole right up there, right? And then we'll talk about where those blood vessels go from there in just a few minutes, right? But we have this structure that forms up here called the glomerulus. Okay. See it over there, right? And you're going to have a lot of these. This is going to happen in the cortex of your kidney. It's going to happen about 1 million times per second. So we're looking at lots and lots and lots of these. In a little while, we're going to get a chance to look at these guys under the microscope. You're going to see that the cortex of your kidney is just jam-packed with little balls of capillaries. And they're really, really easy to find. Okay. Now, we'll talk more about urine formation on Friday, but basically, the way that we start forming urine is that we have to filter our blood. Okay. Have you guys filtered anything today? Do we filter anything else?
Coffee.
What?
Coffee.
Sure, yeah, let's do coffee, right? I'm a big fan of coffee. So yeah, that's probably something that we do filter. Many of us filter kind of every day, right? So when you guys make coffee, one of the things that you have to have is you have to have a coffee filter, right? What's the purpose of that coffee filter?
To filter the coffee.
What? Yeah, no shit. But what is it? But why?
So you don't get the coffee grounds in your
Right. To keep the coffee grounds out of your coffee, right? That's why you use a coffee filter, is to keep the big stuff on one side and to let the other stuff go through, right? So we have to filter our blood in a very similar way, right? We want to keep some stuff in our blood, and we want some stuff to get out, right? What's the big stuff in our blood that we might not want showing up in our urine? Proteins. Okay, good. Yeah, I don't want protein in my urine. That's big. Is there anything else big in my blood that I don't want in my urine? What do we see under the microscope? What do we spend a whole day counting? Blood cells, right? Yeah, we don't want white blood cells. We don't want red blood cells. We don't want those formed elements showing up in our urine either, right? If those things show up in our urine, we've got problems, right? The filter is torn, right? So the filter needs to happen here, and that filter starts with this little ball of capillaries, right? There's a little bit more to it. We'll talk about that on Friday, but suffice it to say, the glomerulus is the filter, right? This is where we're pushing that fluid out of our blood, and we're starting to form urine. Okay. But we have to have something to catch that filtrate with, right? We don't want it to just get filtered out in space. We have to have something to catch it. So what we have sort of surrounding it is we have the beginning of something that we call a renal tubule. Okay? So surrounding this glomerulus is a capsule. Okay? Surrounding it is an epithelial capsule that is going to essentially act as a funnel, and it's going to catch all of that stuff that we're filtering out of our blood. Okay? So we have a capsule right here. It is made up just of epithelium, simple squamous epithelium. We'll look at it under the microscope a little bit more later on today. But this capsule that we see right here is called the Bowman's capsule. It's also called the glomerular capsule. So this is the Bowman's capsule. Or the glomerular capsule. endothelial cells, right? So what that's going to do is it's essentially going to capture the stuff that we're pulling out of our blood or filtering out of our blood, and it's going to funnel it into something called the renal tubule, which is what we're going to start right here. Okay. So the first part of our nephron is what we see up here on the board. Okay, we have the glomerulus. We have the glomerulus plus the Bowman's capsule. We add those two together, we get something called the renal corpuscle. The renal corpuscle is what we have, right? So what we have up here is we have the renal corpuscle. Okay? What we have over here is we have the renal corpuscle. Okay? And then we're going to start the rest of the nephron here in just a second. Any questions so far? We gotta get caught up, and then I'm gonna erase some of those labels just for the sake of clarity, and then we'll take a look at the rest of them. I need bigger boards. I don't have enough board space. Okay. All right. So the renal corpuscle is right here. This is what's capturing that blood that is getting filtered out. This is where the fluid from the urine is coming from. It's very small blood plasma. There's stuff in there. We'll talk about the stuff that ends up in there on Friday, but this is essentially what we have, right? And then what we do is we send that filtrate through this tubule. And the very first part of the tubule that we have up here, this is called the proximal convoluted tubule. Okay. Proximal, because it's close to that renal corpuscle. I'm going to abbreviate it here as PCT. And then I'm going to come over here and I'm going to define that for us. PCT equals to proximal convoluted proximal convoluted tubule, PCT, not to be confused with the Pacific Crest Trail, which is completely different, right? Okay. So proximal convoluted tubule, I'm drawing it here fairly straight, right? But we're going to look at the models in just a couple of minutes, and I have a couple of other PowerPoint slides to show you that show that it really is convoluted. It's very twisty and turny and stuff like that. But for the sake of clarity, we usually draw it fairly straight. I give it a little bit of a wavy line just sort of indicate that it's not straight. But we typically draw it straight. So there's our PCT, proximal convoluted tubule. From there, the renal tubule takes a kind of an interesting turn. It creates this structure that we call the loop of Henle. Okay? So what it does is it kind of takes a sharp dip, okay? And that dip makes a hairpin turn and comes back up immediately. Okay? So we end up with a structure that sort of looks like this. Okay? This is called the loop of Henle that we see right here. I'm just going to go ahead and do this loop of Henle. Now, loop of Henle is over here as well. We have this structure is this. Now that here we can turn. And that is this. And then we have another convoluted tubule. If this was the proximal convoluted tubule, what do you think is next? Distal convoluted tubule. Very good, right? Distal convoluted tubule is next. We usually abbreviate that as the DCT. The
suffice it to say for right now, by the time the blood that's filtered out of here makes it through all of this, by the time it gets over here, it pretty much resembles urine at that point. There's a lot that's happened to it by the time we get over here, but it pretty much resembles urine. And then there's one final tube right here called the collecting duct. That will collect that urine, as the name implies, and then we'll take that urine to eventually out of the kidney. So our collecting duct is kind of over here, and that goes all the way down through the medulla out of the kidney. Okay? So we end up with a structure that kind of looks like so. So this entire thing that we see right here, this entire structure, is called a nephron. Right? This entire thing is called a nephron. And as I mentioned a second ago, each one of your kidneys has about 1 million nephrons in it. So a number of these. But these are the space where urine is formed. Okay? The rest of the kidney is basically there to support these 1 million or so nephrons that we have. Okay? All right. So we still have to talk about how these guys work, but really this is the anatomy of the kidney. All right? So what do you guys have on your benches? You guys have some models.
Everyone else
These are
making soda.
probably one of the better study things. Do we have any questions about this so far? Before I go through some models. Okay. So these models that you guys have on your benches, you guys all should have one of these. There's like 3 vignettes right here. With each one, we're zooming in a little bit further. So let's go ahead and take a look here at what we've got. Whoa. Okay, so the very first vignette that we see right here is the very first vignette that we see here is the entire kidney. Here, have a frontal section of it inside. You can see your blood supply and the way that it branches, and those very small cortical radiate arteries are up here at the very edge. Your renal vein is right here. The ureter is here. One thing that we can see is we can see how the ureter is basically like a big funnel that's collecting urine from all of these smaller funnels, and then that will send the urine to the urinary bladder. Of course, that's how the models are sort of scattered about it. The human kidney is a little bit unique. Those collecting ducts, like we see right here, and like I have drawn on the board over here, are organized into several what we call renal pyramids. So there's a renal pyramid right here, there's a renal pyramid here, here, and so on and so forth. Human kidneys do have multiple renal pyramids. Not every animal does. We'll take a look at a microscope slide in a while. But nevertheless, we have multiple renal pyramids. So on this, we can look at blood supply, we can look at the cortex versus the medulla of the kidney, and so on and so forth. The second one that we have is basically up close of one of these renal pyramids. So we take a look here, we see that renal pyramid. Now we can see the cortex and the medulla right here. You can see multiple examples of nephrons. We have nephrons here, scattered all about. Some of them are complete, some of them they don't show everything, but they're clearly there. This is a collecting duct that we see right here. And we can see how this structure is collecting that urine and dumping it into this funnel, this minor calyx that you see right there. So we have that. And then what we do finally is we blow up one of these structures right here, and we can see that over here, where you have your glomeruli
surrounding it. You have your proximal convoluted tubule right here. You have your afferent and efferent arteriole and so on and so forth right here. So we're kind of taking a step-by-step view, blowing up this structure and looking at it a little bit more closely. Real quick, just let me say one more thing about nephrons, and then I'll shut up for a while and I'll let you guys spend some time with the models, because I want to encourage you guys to do that. One thing about nephrons really quickly is there are two types of nephrons. You guys couldn't see me awkwardly looking through my slides. There are two types of nephrons. The two types of nephrons that we have are what we call juxtamedullary and cortical nephrons. And really, they're named based on the length of the loop of Henle. Okay? Cortical nephrons have a loop of Henle that stays almost entirely within the cortex of the kidney. Juxtamedullary nephrons have a really long loop of Henle that dips down into the medulla. So I have a couple of pictures here. Let me show you these pictures. I think this one works pretty well. So here we have two examples of these. In the one on the left, you'll notice how this loop of Henle on this nephron doesn't dip below that little border that we have between the cortex and the medulla, right? So that's what we call a cortical nephron. The one on the right, however, you'll notice that this one has a much longer loop of Henle that goes down into the medulla before it comes back up, right? That's what we call a juxtamedullary. Juxtamedullary means next to the medulla. So it's down into the medulla. In our kidneys, we have both types. We have both cortical and juxtamedullary nephrons. However, the vast majority of nephrons that we have are cortical. About 80% of our nephrons are cortical nephrons. Okay? Now, 80% of our nephrons are cortical, about 20% are juxtamedullary. We'll talk about the implications of that on Friday, but this little teaser right here, kangaroo rats, our little friends, have about the opposite. They have about 80% juxtamedullary nephrons and about 20% cortical. So you see a difference in the distribution of the nephrons in terms of the length of those loops. Okay? So those are a couple of things that we need to pay attention to. Just real quick, just to kind of show you this. This is what a nephron actually looks like. It's more closely related to what you guys have on your models. You'll notice that those convoluted tubules are convoluted, right? You have your Bowman's capsule is right here, wrapping around that glomerulus, and then that proximal convoluted tubule kind of comes up here and it wraps all the way around, right, like this. And then we dip into our loop of Henle right here. So we have our descending limb of our loop of Henle right here. We have our ascending limb right here. And then we have our distal convoluted tubule, which, again, as the name implies, is very twist and turn. And then we dump as far left and right there. And this is probably more reminiscent of what an actual nephron looks like, as opposed to what we've drawn on here. It just makes a little bit more sense if you sort of straighten out those convoluted. The other thing that I want to point out really quick before I shut up is that you'll notice that efferent arteriole, weaves right here, and then it turns, and it actually turns into a second set of capillaries that completely surrounds this entire thing. So this entire thing is going to be completely wrapped in capillaries. I'm not a good enough artist to draw that, so I chose not to. I also don't want to confuse the drawing by adding more stuff to it. But there is a very close association between the fluid that we find inside of here and then the blood supply that's going to be drawn out. Right? When we've seen one thing in this unit, we've seen that we're moving our blood closer and closer to different spaces. And again, that's where the action happens. That's where movement occurs. And we'll take a look at that a little bit more. These are called your paratubular capillaries because they're next to the tube, which is there.
So why don't you guys spend, I'm going to shut up for a little while. Why don't you guys spend a little bit of time with these models? Let's take a look at your lab really quick and let you guys go through some of the structures that are listed in your lab. We'll definitely talk about a few of these, and that's totally fine. But I'll let you guys explore for a bit. We'll kind of go from there. So in our lab, you guys have the external gross anatomy of the kidney. We have the internal gross anatomy. We have the nephron structure, which we've already talked about a lot. And then we have the blood supply and stuff like that associated with it. So why don't you guys spend some time in part A of your lab and the models, and then start to identify some of these features that are. Proximal convoluted tubule. I follow that around, and when I get to this is my descending limb that we see right here. When I get down to the hairpin down there, that's my ascending limb that we see right there. Go through my distal tubule and then ultimately to my collecting duct, right? Again, they're color-coded, but you can do the same thing here, right? You can trace that proximal tubule all the way around until you get to your descending limb and so on and so forth. Keep in mind that on a lot of these models, when we have them on opposite sides of the collecting duct, they'll be like this big of each other, right? But if you just trace, that's kind of the easiest way to sort of tell the difference between the descending limb and the descending limb. Another one is about the types of nephrons. I think that the models that you guys have at your benches, the big ones, don't really do a good job of differentiating football and also not all in the cortex either. I suppose we could go into a long philosophical discussion about what makes a cortical nephron a cortical nephron, but probably wouldn't be curious, right? So, which is fine. So I'll probably just avoid those ambiguities on the test. If I ask you to identify a cortical nephron, it'll be like this. If I ask you to identify a juxtanephron, we'll save the philosophical discussions for this. So we can kind of do that, right? So we have those. This, I think, the big models you guys have inches and this little part right here, I think do a really good job of showing those capillaries that surround the entire nephron. Down here in the loop of Henle, those capillaries, those paratubular capillaries, they're called the vasa recta. Okay, we'll talk about the significance of that vasa recta. Really cool system. Over here, we see a few other things, and these were things that I was specifically asked questions about by a few different groups, which I totally expected. The first thing is about podocytes. A couple of groups asked about those. That name, or that word, podocyte, if we think about what it means, that prefix podo means foot, right? A pod is a foot. And cyte, of course, means cell. So that word, podocyte, literally means foot cell. These capillaries, this glomerulus, is covered with these cells that are called podocytes. On Friday, we'll talk a little bit more about their significance. But suffice it to say, what they essentially do is the foot processes of those cells create slits that essentially add to the filter that you get when you start to eat. So these foot processes of these podocytes are going to add to that. This model actually shows these podocytes and their foot processes fairly well. Another question that we're usually asked is which one is the afferent and which one is the efferent arteriole. We see that there are indeed two of them up here. Generally speaking, the afferent arteriole tends to be a little bit bigger than the efferent arteriole. So we see that this one is just a tiniest bit bigger than the one next to it. So that makes this one the afferent arteriole. And then finally, the last question that we're often asked about is, about the juxtaglomerular apparatus that's listed in there now, right? There's a lot of big words there, right? Juxtaglomerular apparatus. It sounds like it should be a band name, but it's not. It's just a part of the kidney. So the juxtaglomerular apparatus consists of two groups of cells. Okay? First of all, what tube is this right here? This is the proximal convoluted tubule, but what tube is this right here? It's a what? I think I heard that. I'm just deaf. Distal. Why are you whispering? Say it like you own it. Own it, man, own it. Distal convoluted tubule, right? Yeah, it is. So if we take a look over here, right? If we take a look over here and you look at any one of these, let's take this one for example. You know, you've got your proximal convoluted tubule like right here, but then look where that distal convoluted tubule passes. It's like right next to the glomerulus. We see that happening here. We see that happening right here, right? So we see it right here. We see it when we cut this section. We see that we have that distal convoluted tubule right there. This isn't just an axon. Right? That distal convoluted tubule has to pass through the glomerulus because of this thing called the juxtaglomerular. So there's a couple of aspects to that juxtaglomerular apparatus. The first one, and I wish it was color-coded a little bit better on these models, but there's this group of cells that's kind of right here in the distal convoluted tubule. Those are called macula densa cells. They're called macula densa cells. And then in our afferent arteriole, there's cells as well right here they're essentially going to work to control the flow of fluid through the glomerulus. Okay? If the flow gets too high, what we can do is we can turn it down, because the distal convoluted tubule will say, oh no, there's too much fluid flowing through here. It isn't. But it will essentially say that. And then those macula densa cells will send messages to these granular cells, and they'll cause that afferent arteriole to constrict, and it will turn down the flow through that arteriole, and it will turn down the flow through the when the flow gets too low, what happens is this arteriole will relax, and the flow will increase through this. So this allows us to regulate our rate of filtration. We'll talk a little bit more about that. But I know that I get asked that a lot. Where are the macula densa cells? Where are the granular cells? And it's just this very close proximity between these two groups of cells that allows them to. Okay. Any questions about anything else? I feel like those are usually the any others come up, we can definitely talk about it. But let's really quick. Let's take a look. Some of the time that we have left today. Let's take a look at the kidney under the microscope. In just a second, I'm going to ask you guys to go back to the microscope slide of the kidney. There are a couple of different examples back there. These are two of them. I don't normally show the slide right away, but these are a couple of the options. There's a couple of others floating around. Pulmonary arteries and veins, and then those primary bronchi enter the lung as well. Right? That indentation is called the hilum of the kidney, or in the case of the lung, the hilum of the lung. Okay? So we can see that hilum right here, where that tissue becomes very indented, and then we come back around, and then we see that it becomes very circular again, kind of all the way around. One thing you can see on this slide, on this side, is we can see this kind of wavy line up here on top. That's that renal fascia that becomes separated from the kidney itself. So we can actually see that renal fascia up here, that renal capsule that's kind of separating from the kidney itself. In the renal hilum on this one, you can see this guy, right? This guy right here, okay, that's that single renal papilla from that single renal pyramid this kidney has. If it had multiple, of course, it would be a lot shorter, but that's that single one that we have right there, this single renal pyramid that exists. So that's that renal papilla, that very bottom of that renal pyramid that exists right there. You can definitely see that here as well. Okay. And then finally, the last thing that we see is you can see that around the kidney, especially right here in that area around the hilum, is a whole bunch of adipose tissue, right? You can't necessarily see that on all of our slides, but around this area of the kidney, all of this is adipose tissue. One of the things that was on your list of structures to find was the pararenal fat capsule. That's what all this adipose tissue is, right? There's a lot of adipose tissue that surrounds the kidney. And it's hard to see on our models, but we do see it here on this guy as well, under the microscope. Okay. So, with all of that said, what we need to do is we need to go to the kidney itself, and we need to start to look at a couple of things. And the first thing that I think we should do is we should identify the difference between the cortex and the medulla of our kidney, right? So, let's come back to our model real quick. Wish the floor was stickier, I could move quicker. I would never get away from like somebody on this floor, I don't think. It would be like powerful. But let's come back to our model real quick. So right here, this is a good example of kind of what we're looking at. Of course,
this would be a single renal pyramid in the kidney that I have up on my microscope slide. But when we look at the medulla, okay, remember the medulla is going to be made up almost entirely of collecting ducts and then those limbs of the loop of Henle, right? So the entirety of the medulla is going to be made up basically of tubes that we're looking at in cross section, right? Remember, there's about 1 million of these per kidney, so they're going to be really jam-packed in. Most of them are taken out of here for the sake of clarity, but if this was realistic, the entirety of what we'd be looking at here is all of these tubes jam-packed in here all together. Okay. In the cortex, we see tubes. We see our proximal and our distal convoluted tubules, right? We see those tubes that are in here, but we're also going to see the actual renal corpuscles, right? The glomeruli and the capsules that go in. The good thing is these are really easy to pick out of this. That's going to make our job full. So let's go back to our slide and let's look to that area where we see these capsules, and then look to the area where it turns to more tubes. Right, so up here, they see, I look here, and it just looks like tube cross section. But up here, the tissue gets a lot more solid, and I see a lot of these guys. All of these are going to be our glomeruli and Bowman's capsules, right? There's 3 more right there. There's another one right here. There's 2 more right there. There's another one there. Even a couple down in here, kind of like this. 2 more strays kind of down in there, right? So when I kind of step back and I look at this tissue under the microscope, I see, generally speaking, I see a dividing line. I have to stand back. I can't stand right at the screen. Right about here. Okay. You guys see that dividing line that I'm looking at right there? And I drew it, so obviously you see it. But do you see why I drew it where I drew it? How that tissue kind of changes character. There are a couple of glomeruli down below my line. It's not a hard line, okay? But it's pretty darn close. So that makes this my cortex, and that makes this area, generally speaking, my medulla. Right? So the very first thing that we should do on our slides right now is let's identify the difference between the renal cortex and the renal medulla. Villi on the luminal side. Okay, what that does is it does increase the surface area of the cells, and we'll talk about the implications of it on Friday. We'll notice that by the time we get to the distal convoluted tubule, those microvilli have disappeared. They're no longer there. Okay, so what that's going to do is it's going to make the distal convoluted tubule look a lot more open. Okay. Good one. This is probably a proximal tubule here, where there's just very little lumen present that looks very occluded.